Welcome to PK's classes. Namaskar. Today we are going to study about the polyclonal antibodies. Polyclonal antibodies. So first antibody. Antibody is a substance produced by our body in response to one antigen. And antigen on its surface has epitopes. And antibody on its surface has paratopes. Antigen epitope and antibodies paratope, they interact. So the interaction leads to neutralization of the antigen. Suppose this is a virus. So this virus on its surface has these epitopes and the antibody can bind to a specific epitope. So when we have a mixture of antibodies which can bind to different epitopes. Suppose say this is one antibody. Antibodies are Y-shaped structures. So this is another antibody. So this is another antibody. So this is another antibody. So this mixture of antibodies, we call them as polyclonal antibodies, which can target different epitopes on the antigen. So antibodies are produced by the B lymphocytes or B cells, and as you know, the B cells are of two types, plasma B cells and memory B cells. The plasma B cells produce antibodies. So when we have a mixture of B lymphocytes, then those B lymphocytes will produce a mixture of antibodies which can target different epitopes. Then that mixture is called the polyclonal antibodies. So poly means many and clonal means a mixture. Poly means many, so many clones of B lymphocytes. So we can say many clones of B lymphocytes. So each producing one antibody with affinity to specific epitope of the same antigen. Then we call them as polyclonal antibodies. So multiple clones of B lymphocytes produce a mixture of antibodies or immunoglobulins, and that mixture is called polyclonal antibody. Suppose say this is the antigen. This is the antigen. So against this antigen, so when this antigen is introduced into the body of one animal, the animal will produce different antibodies because each type of B lymphocyte produces a different antibody. And when we have these multiple clones of B lymphocytes, we have a mixture of antibodies, say suppose antibody 1, antibody 2, antibody 3, antibody 4, each targeting a specific antigen. But each of them will bind to the different epitopes in the same antigen. Suppose this antigen, suppose this is the antigen. In this antigen, suppose this is epitope 1, epitope 2, epitope 3, epitope 4. So the mixture of antibodies, they can bind to a specific epitope. Let us see here. Suppose this is the antigen. This is the antigen. And this antigen has having three epitopes. Epitope 1, epitope 2, epitope 3. And one antibody 1 targeting this epitope. So antibody 3 targeting this epitope, antibody 2 targeting this epitope. So in this case, this mixture of antibodies is called polyclonal antibodies. Okay. So now I think it's clear. So in response to one antigen, our B lymphocytes, because we have the multiple B cell lineage, each B cell lineage producing a specific antibody which can target to a specific epitope. Suppose different epitopes are there. Suppose this is one epitope, this is one epitope, this is one epitope, this is one epitope. So we will have a mixture of antibodies which can target to different epitopes in the same antigen. So that mixture is called the polyclonal antibodies. So though this mixture will have no less specificity because it can bind to different types of epitopes, but we can say they are broad in action. They can bind to a variety of epitopes in the same antigen. So the method of production, we have to first inject the antigen into the animal, say rabbit. Then this antigen activates the B lymphocytes in the rabbit, and these B cells or B lymphocytes will proliferate. So we'll have two types of B cells, memory B cells and plasma B cells. And the plasma B cells will secrete the antibodies which are present now in the blood. So we can take the serum and from the serum we can separate the polyclonal antibodies. So what happens when we produce these polyclonal antibodies? In addition to the antibodies against the antigen, it also have antibodies that do not recognize the antigen. So all the antibodies in the serum we collect. So from that we have to collect antigen specific antibodies. So we need purification. And after the purification we will get purified antigen specific polyclonal antibodies. So to simplify, so we can say that first we have to immunize the animal, then collect the blood, purify, we'll get the polyclonal antibodies, which is a mixture of antibodies with different epitope targets on the same antigen. Suppose this is a microorganism with antigen, and it has say on its antigen surface it has say 4 epitopes, 1, 2, 3, 4. Accordingly, we will have a mixture of antibodies targeting each epitope. That mixture is called as polyclonal antibody. But when we say for one epitope we produce the antibody, that antibody will be, this one is monoclonal. So this is the difference between the polyclonal and monoclonal. So in polyclonal we have the multiple B cell lineage, and in monoclonal we have single B cell lineage. So only one B cell we will collect, and then we will proliferate them. Suppose this one B cell we suppose select, and this B cell we will proliferate, we will make clones, and then if we produce, then we will have monoclonal antibodies. But when there is no differentiation of all these B lymphocytes, then we will have a mixture of antibodies, and that mixture of antibodies is called polyclonal antibodies. So I think now it's clear. You can see here, suppose this is the antigen, epitope 1, epitope 2, epitope 3. Accordingly, we have the antibodies against those epitopes, and this mixture of antibodies is called as polyclonal antibodies. Whereas if only against one target epitope we are producing antibody, then this antibody will be monoclonal antibodies. So a difference number of species we can use for the production of polyclonal antibodies, say sheep, goat, rabbit, mice. So all these animals we can use. So now we can define, and you can note down, polyclonal antibodies are the antibodies produced by multiple clones of B lymphocytes in response to a particular antigen. In other words, polyclonal antibodies are a mixed population of antibodies or a mixture of immunoglobulins where each antibody recognizes different epitopes of the same antigen. Then, in other words, we can also define polyclonal antibodies is a mixture of neutralizing antibodies that are found in serum of an animal species in response to a particular antigen. Or you can make a mixture of all these definitions. So what are the characteristics of polyclonal antibodies? They bind to a variety of epitopes in an antigen. Suppose there is a small change in the protein or antigen structure, then also it can recognize because it has a mixture of antibodies. And because of this, there is one disadvantage that it has cross reactivity. With similar antigens it can react. So it is a heterogeneous antibody population. It is less specific because it can bind to a variety of epitopes, and because of this, there is increased likelihood for cross reactivity for similar antigens. So we cannot say that this antibody will bind to the specific antigen. If there is similar antigen available, it may also bind. This is the disadvantage with polyclonal antibodies. So you can write the advantages and disadvantages. Before that, this shows the production. This is one in vivo method. We have to use animals, different animal species are required, and then we have to immunize those animals. Then we collect the serum, and mostly the antibodies are produced in a large scale using the horse. Then after collecting the serum, we have to purify for the collecting the antibodies against that specific antigen. So this is relatively easy technology as compared to the production of monoclonal antibodies, and fewer skills are required as compared to the production of monoclonal antibodies. So the advantages are it is cost effective, cheap to produce, it has a short production time, only 2 to 3 months required. So this short production time, this is as compared to monoclonal antibodies. So many host species options are there. So all these advantages are as compared to monoclonal antibodies. Fewer skills are required for production, relatively easy technology, and it has high affinity because it binds to a variety of epitopes. That's why we call it as high affinity. But specificity is low because it is not specific to a specific epitope of an antigen. So tolerance of minor changes, this is one advantage because because it can bind to similar antigens also. So if a certain change in the structure of the antigen also is there, then it may also bind. So the disadvantages are it is less specific, and batch consistency is low because lot to lot variability will be there. Different batches vary in composition. So suppose today we have produced a batch, the composition, and next batch we produce 3 days after, that batch will have a different composition. We cannot get the same composition every time with polyclonal antibody, but in monoclonal antibody you can get that. So disadvantage another is increased likelihood of cross reactivity for similar antigens, and already I have discussed. So this can be used mainly as antivenom preparations, in diagnostics, in general research applications. So these are all the applications of polyclonal antibodies. Thank you.